Hello everyone, my name is Pallavi and in today's class we are going to continue with the chapter Molecular Basis of Inheritance. So in the previous class we discussed that the nucleic acid is the genetic material in the living organisms and the nucleic acid can be DNA or RNA, and a nucleic acid is a polymer of nucleotide. So we discussed the structure of nucleotide. We also discussed the structure of DNA, which was given by Watson and Crick, and then we discussed the silent features of this double helix model, and we concluded that the length of the DNA is 2.2 meters, which is very long, and the dimension of a nucleus in a mammalian cell is 10 raised to power minus 6 meters, which is very small. So how such a long DNA is going to pack itself inside the nucleus? Today we are going to discuss about that and the different experiments which were conducted to find out that which is the genetic material in a living organism. Before starting with the packaging of DNA, I just want to discuss about the central dogma. Central dogma was given by Francis Crick. So he gave the flow that how a DNA is going to express itself and it is going to form proteins. So DNA first replicates itself. So it is DNA replication. After that, the DNA converts into messenger RNA. Messenger RNA along with the ribosomes is going to synthesize the proteins. So the conversion of DNA into mRNA is called as transcription. And then mRNA will produce protein with the process of translation. But what about the viruses? Viruses have RNA, and from that RNA they sometimes form DNA. So what kind of process they are going to perform? They perform reverse transcription. So I can say that in viruses a reverse central dogma is being conducted. Reverse central dogma. So that was central dogma, and in the further chapter when we are going to discuss it, so we have to study about each and every process in detail, the DNA replication, transcription, translation as well. So before coming to these major topics, we have to discuss the packaging of DNA. So how do we calculated the length of the DNA? Let's just quickly recapitulate. So the length, to find the length of a DNA of a mammalian cell, mammalian cell, what we did is we just took the total number of base pair present in the cell and we multiplied it with the distance between the two consecutive base pairs. So what was the total number of base pair in a mammalian cell? It was 6.6 into 10 raised to power 9 base pairs. And what was the distance between the two consecutive base pair? It was 0.34 into 10 raised to power minus 9 meter per base pair. So this cancels out and the answer will be 2.2 meter. Simple. Now in NCERT, we are given with the length of the E. coli DNA and we have to find the total number of base pair present in it. So how we are going to calculate? So the length of DNA in E. coli cell, it is given 1.36 ml. So first we have to convert this millimeter in meters. So what I'm going to do is 1.36 into 10 raised to power minus 3 meter, simple. and I have to find the total number of base pairs present in it. So again, the formula remains the same. To find the total number of base pair, we are going to divide the length of the DNA with the distance between the two consecutive base pair. So to find the total number of base pairs, we are going to divide the length of DNA with the distance between the two consecutive base pairs. We already know the values. What is the length of the DNA? 1.36 into 10 raised to power minus 3. And what is the distance between the two consecutive base pair? It remains the same. 0.34 into 10 raised to power minus 9. So it comes out to be 4 into 10 raised to power 6 base pairs. Easy. So these are the number of base pairs present in an E. coli. Let's just find out how the packaging of DNA helix is done in prokaryotes, because E. coli is a prokaryote, and in eukaryotes, in mammalian cell. So what about the packaging of DNA in a prokaryote? So in prokaryotes, the DNA, there is a double-stranded DNA. So I'll draw a double-stranded DNA. So this is a double-stranded DNA. And we are aware that prokaryotes don't have nucleus. They have nucleoid. So they don't have proper nuclear membrane and the DNA is present in a naked form. DNA, nucleoid consists of DNA which is negatively charged. DNA is coiled with few positively charged proteins. So here if I will draw, there will be a few beads of proteins around which the double-stranded DNA is coiled. So this is the simple packaging of DNA in prokaryotes. But what about the eukaryotes? In eukaryotes, so packaging in eukaryotes. So eukaryotes have a specific nucleus which is having a dimension of 10 raised to power minus 6, and we have to pack such a long DNA inside it. So we opt for coiling of DNA. So this is DNA which is negatively charged. Why it is negatively charged? Because it has phosphoric acid. So it is having negative charge because of phosphoric acid. And this DNA which is negatively charged, it is going to coil itself around a protein which is positively charged. So here histone protein is present. Histone protein. And why the histone protein is positively charged? Because it has the residues, high level of residues of basic amino acids, which is lysine and arginine. So here the DNA is coiling itself around the histone proteins. Here this is a 2D structure. So in total there are 8 histone proteins which are present. So it forms a histone octamer. And how many types of histone proteins are present? 2H2A proteins, histone proteins, 2H2B histone proteins, and then 2 beads of H3 histones, and there will be 2 H4 histone protein will be present. So it forms a histone octamer. And along with this histone octamer plus this coiled DNA, they forms a nucleosome. They form a nucleosome. Apart from these histone octamer proteins, there is one more histone protein which is called H1 histone. This is the linker histone. So it will also be present in the nucleosome. So I can say that nucleosome is the repeating unit of a chromatin material. So here I'll write nucleosome. It is the repeating unit of chromatin. Now if I have drawn it like this, now I haven't drawn the separate histone protein. So this is a nucleosome, this is a nucleosome. Many nucleosomes, they are going to tightly pack themselves, and it appears a bead on string like structure. Bead on string like structure. And if I'll say what are the beads, beads will be of nucleosome, and the string is DNA. So at higher level, this chromatin, higher level, the chromatin is tightly packed. At this stage, we can see the non-histone chromosomal proteins, which are NHC protein. So they are visible at the higher level of chromatin packaging. And again, at the during the cell cycle or cell division, during the cell division, they are going to condense themselves and they are going to form a X-shaped structure, which is called as the chromosome. Chromosomes are best visible at a metaphase stage. So I can easily see the chromosome during the cell division at the metaphase stage. So this is how the packaging of DNA is done in eukaryotes. Chromatin is of two types. One is heterochromatin, the other one is euchromatin. See the loosely packed chromatin, this region, this is loosely packed. So this will be euchromatin, and the densely packed region is heterochromatin. So if I'll write the difference between heterochromatin and euchromatin, first will be that heterochromatin are densely packed. So if anything is densely packed, so it is going to catch more stain. So they are the dark stained part. Euchromatin is loosely packed. So they are lightly stained. See, as they are loose, they can provide the space for the transcription factors to bind themselves. So the transcription factors. I hope you know what is transcription. We discussed during the central dogma when the DNA is going to convert itself in mRNA. So transcription factors can bind. But heterochromatin is very dense, so it cannot provide any space to the transcription factor to bind. So they don't have any site for the binding of transcription factors. So if the transcription factors are not able to bind, so they cannot perform transcription. So I can write that they are transcriptionally inactive, whereas the transcription factors can bind with the euchromatin sites. So they are transcriptionally active. So this is the difference between chromatin. Now we have to discuss the various experiments which were performed to find out, to search that what is the exact genetic material. See, in 1869, Frederick Mischer was the first scientist to isolate the DNA from the nucleus of a pus cell, and he named it as nuclein. Later, Altman discovered the acidic nature of the nucleic acid, the DNA specifically. So by 1926, the quest to determine the inheritance of genetic material reached to the molecular level. The previous discoveries of Mendel, T.H. Morgan, Walter Sutton, they confined the research to the chromosomal part, that yes, the genetic material will be found inside the nucleus, specifically in the chromosome. But what exact molecule is being inherited from one generation to another as the genetic material was not found. It can be the DNA, it can be the protein, it was not identified by that time. So different experiments were performed to search the genetic material. So the first experiment was performed by Griffith, and we named it as Griffith's transforming principle. It was done in 1922. He experimented on a bacteria which is Streptococcus pneumoniae, which causes pneumonia, and this bacteria has two strains, two different types of strains. One is the S strain. S strain formed smooth, shiny colonies. Why? Because it has a smooth coat of polysaccharide. See, because of this polysaccharide coating, it is protected from the host immune system. So it can easily cause the disease, pneumonia. Whereas the R stained formed rough colonies because they didn't had the polysaccharide coating, they lacked the polysaccharide coat, so they were exposed to the host immune system, so they were the non-virulents, they didn't cause the pneumonia. So what he did is he took the different strains and he injected them in a mice. He injected them in mice. So when S strain was injected in a mice, it got diseased and the mice was killed. Mice was killed. It didn't survive. When R strain was injected in the mice, it survived because R strain was a non-virulent strain and it it don't cause the pneumonia, so the mice survived. After that, what he did is he heat killed the S strain bacterium and again injected in the mice. Now, surprisingly, the mice survived. After that, along with the heat killed S strain, he also injected a living R strain and he saw that the mice was not able to survive. So from these different conditions, what was the conclusion? So from that experiment, we can conclude that there can be some transforming principles that were transferred from that heat killed S strain of bacterium into the living R strain bacterium, and that R strain bacterium got transferred to S strain and became a virulent one, and that's why the mice was not able to survive. But still, this experiment don't give us the molecular basis of the genetic material. So to find the chemical nature of Griffith's transforming principle, in 1933 till 1944, Avery, MacLeod and McCarty, they performed experiment. What they did is to check which chemical transforms R cells two S cells. They just isolated, they extracted all the molecules, protein, DNA and RNA, which are present inside the nucleus. And what they did is, they first digested protein by protease enzyme and RNA by RNA enzymes. And they stopped the supply of protein and RNA by doing this in the R cells. And later on, they saw that the R cells still transforms in S cells and becomes a virulent strain. Whereas when DNA was digested by DNA's enzyme, so the supply, it stopped the DNA to reach to the R cells. And surprisingly, the R cells, the R strain of bacterium didn't transform at it, and it remained non-virulent. So from this, they concluded that yes, the DNA is the genetic material. But still many scientists were not convinced. So in 1952, two scientists, Hershey and Chase, performed an experiment on a bacteriophage. Phage and E. coli. What is a bacteriophage? The virus which causes infection to a bacteria. So bacteriophage is a virus which is going to cause the infection to the bacteria. They have taken the bacteria as E. coli. What they did is, they just grow some viruses in radioactive phosphorus. So the DNA contains, the genetic material contains what? Phosphorus. So it will get attached, tagged to the DNA, and it will make the DNA as radioactive P32 DNA. And they grew few viruses in radioactive sulfur rich medium S35, and the protein coat contained, this is the protein coat, so it contained sulfur. So the sulfur, radioactive sulfur, got tagged with the protein coat and it formed radioactive S35 protein. So they performed the experiment in 3 steps. The first step was infection. First step is infection. So what they did is, they took the radioactive sulfur tagged bacteriophage, which was attached with the protein coat, and the bacteriophages which were radioactive phosphorus, which were having radioactive phosphorus DNA, and they allowed them to infect the E. coli. So these are the E. coli cells, they infected. And what they are going to do, they are going to infect and transfer their genetic material inside the E. coli bacterium. So they are going to transfer their genetic material inside the E. coli cell. After that, they blended all the solution. So second step was blending, and that's why we refer this experiment as blender's experiment. So E. coli are gently agitated by a blender, and the viral coats are removed from the bacterium. So these viral coatings will be removed, separated from the surface of the E. coli. The bacterial coating will be now present in only in the suspensions. After that, the third step was centrifugation. Centrifugation. So the culture was centrifugated to separate the bacteria and the virus particles. After that, they studied the solution, the supernatant as well as the bacterium. So when the bacteria was infected by a bacteriophage which was having radioactive sulfur protein coat, what they saw is inside the E. coli there was no radioactive element present. So there was no sulfur radioactive sulfur present, and in the supernatant they detected that radioactive sulfur is there. So it showed that the protein coat is not acting as the genetic material. Whereas when they took the bacteriophage, which was having the phosphorus, radioactive phosphorus, which was tagged with the DNA, what they found is in the E. coli, the radioactive phosphorus was present. And in the supernatant, there was no radioactive part present. So it just confirmed that the DNA was the genetic material which is going to pass from the bacteriophage to the E. coli cells. And this experiments gave the final and the convincing proof that the DNA is the genetic material in the living organisms. So this was the class for today, and you can revise everything from my handwritten notes and you can download it from the description. We'll continue with the chapter in the further classes. Thank you.